Pancreatic cancer currently has a 5-year survival rate of less than 3%. The lack of knowledge of the molecular determinants involved in the pathogenesis, proliferation, and maintenance of this tumor type are factors that contribute to this disease's poor prognosis.
The hedgehog signaling pathway is an essential pathway active during pancreatic embryonic development. The misregulation of this pathway has been seen in many forms of cancer, including pancreatic ductal adenocarcinoma, or PDAC. Researchers at Mass General and Harvard Medical School sought to determine the importance of the hedgehog pathway and its role in the development and maintenance of PDAC.
First, the researchers had to prove that this pathway was being misexpressed. This was done by observing sonic hedgehog, a secreted hedgehog ligand that triggers the hedgehog pathway, using in situ hybridization and immunohistochemistry of 20 human pancreatic tissue specimens. In normal adult human pancreata, no sonic hedgehog was detected. However, evaluation of pancreata from patients with adenocarcinoma revealed sonic hedgehog expression in 70% of specimens.
Additionally, the researchers noticed that precancerous lesions of all stages were expressing sonic hedgehog. This suggested that hedgehog signaling plays a crucial role in pancreatic cancer from its early formation to its proliferation and maintenance. To test this, the researchers analyzed the pancreata from 4 transgenic mice with misexpression of sonic hedgehog, driven by the pancreatic-specific promoter PBX1. Histological analysis showed that all 4 mice had developed significant phenotypic changes similar to those seen in the precancerous lesions in human PDAC progression. Further analysis revealed that these mice overexpressed HER2, NEU, and contained KRAS mutations, 2 genetic alterations also seen in the human progression model.
They also found that smoothened, a hedgehog-associated protein, was expressed through both an autocrine and a paracrine mechanism of sonic hedgehog. Next, Laizure and her team wanted to determine whether PDAC cells require the sonic hedgehog pathway in order to proliferate and survive. They used cyclopamine, a drug which directly interferes with smoothen, to stop hedgehog signaling in order to gather both qualitative and quantitative data.
After exposing 3 pancreatic cell lines to cyclopamine, the scientists observed a 2.5 to 3.5-fold increase of apoptosis and a 75 to 85% reduction of proliferation in cells, but determined that cyclopamine was only effective on 50% of cell lines.
As a more definitive test of the ability of cyclopamine to inhibit tumor cell proliferation, the researchers developed a secondary treatment model where pancreatic cell lines were injected subcutaneously into immunocompromised mice. In the mice treated with cyclopamine, researchers saw a 50 to 60% decrease in tumor mass compared to the control. This demonstrates that hedgehog signaling is essential for the proliferation and survival of PDAC, both in vitro and in vivo. Future studies will be needed in order to assess the role of hedgehog activity in stem cell-mediated tissue regeneration and PDAC. Identification of a role for the hedgehog pathway in the initiation and maintenance of pancreatic cancer suggests that this pathway may hold promise for new diagnostic and therapeutic approaches.